Hi, my name's Tom McHugh and I'm the team leader for the Lab for Circuit and Behavioral Physiology here at Riken CBS. And today we're going to make a short video with the help of some of my staff to introduce our lab and what we're interested in and show you around a little bit so you can experience a visit to campus even virtually today. So we're now in my office and I'll tell you a little bit about what our lab studies. Our primary interest is memory and how the circuits in our brain allow us to encode, consolidate, and recall memories. And specifically, we focus on a part of the brain called the hippocampus, or in Nihongo, kaiba, which is part of the medial temporal lobe. And it's a part that we know is crucial in humans to remember the experiences or episodes that make up our lives, the who, what, where, when memories that we can easily recall and share. But we don't study humans, we study primarily mice, and we choose mice as our model system because we know a lot about the hippocampal circuits in those animals, and we have very well developed both genetic and physiological approaches that allow us to manipulate activity of neurons in that part of the brain and also record their activity and see how our changes affect how the neurons underlie the memories that form in these animals' heads. So I'm going to put on my mask now and go out into the lab and show you a little bit about how we do what I just described. Alright, so we're in one of the experimental rooms now, and I'm going to show you a little bit about what we do. Introduce you to postdoctoral research in the lab, Vlad. Hello. And so I'll show you the screen very quickly, as you see. And what he's doing here is listening to activity in the hippocampus of a mouse. Here is what we call local field potential, which is kind of population oscillations that tell us a lot about how information is being processed and can tell you, just by looking at the screen, the state the mouse is in. And over here we're looking at spiking activity of a single neuron. You can hear that little crackle in the background. Each one of those little sounds is a spike coming from a neuron in the hippocampus of the animal. Now you can see our little friend here. He has these bright lights in his head so he can track his position in the maze. These neurons code for specific places in this location. And the little cap on his head contains recording probes that we can lower and raise to record cells in this area of the brain. Alright, now I'm in the lab. I'd like to introduce one of my graduate students. I'll flip the camera around and let her introduce herself.
Hey everyone, I'm Hefei. I'm a PhD student who stayed in the lab for several years. And then recently I'm writing my the thesis for my PhD project, which is related to the importance of CA3 on the lateralization of CA1 with the in vivo electrophysiological recording technique. So this is what I'm doing. So here shows that this is one of the tetraodes that we record during when mouse is freely moving. Here the different colors shows different cells. And then this is how it looks like after clustering. So after we manually cluster all the cells from the tetraode, and then we put the data in the MATLAB and then do analysis. Yeah, I like the lab, although recently it's a bit boring because not many people stay around me. But still I miss the time that when the lab is busy. I hope we can get back to that situation soon.
Alright, and now I'd like to introduce you to another student in the lab. So I'm going to flip the camera around once more.
My name is H. Toyoizumi. I'm a graduate student at Todai as well as here at Riken CBS. I study memory and consciousness, basically. As you can tell, Tom, our boss, is really nice, and everyone in the lab is pretty friendly. So, I mean, if you do decide to come to this lab, you won't regret it.
Thanks.